So these are some notes on the fruit fly eye color network because some people asked about them yesterday in the real-time meetings, and I realized that I actually had pictures of the data from when we used to do this lab in class. We haven't done this lab for a couple of years because we replaced it with the C. elegans, but I still have the data because it was pictorial. And so first I just want to show you that fruit flies do actually have different color eyes, and you can see that under a regular dissecting microscope. So this eye here, this is what's called wild type, and that's when all the enzymes are functional. And then the other colors of eyes that we were looking at all have to do with non-functional proteins. So, and we're going to look at that in the next slide. But the white, so this is the white eye color, and that has no pigments. This is the scarlet, it's kind of a brighter red, and it has no brown pigment. And then this is the sepia, and it has no orange pigment. Okay, so that's where you can actually see what these look like. And what we used to do in class is I would give you one of each of these flies and teach you how to lop its little head off and mash out the eye pigment, and then we used a process called chromatography. So some of you have used chromatography before, where you put like a little dot of a black marker on a piece of paper, and then you let the water kind of draw up the piece of paper, and it separates the different pigments that are in your black marker. You use different solvents for different things. So like a Sharpie, that won't work for if you're using water as your solvent, because Sharpies are not soluble, cannot dissolve in water, so you use like rubbing alcohol works if you want to separate different colors of Sharpies. If anyone's interested in doing that at home, if you've never seen it, I can give you some quick directions. Just email me and I'll let you know exactly how to do that. In this case, we used a solvent that was equal parts ammonia and equal parts rubbing alcohol, and that was our solvent for the eye pigments. So this is now the data and the pathway. So one of these two was on your assignment. So if you did the mainstream assignment, I gave you the pathway and then you had to derive the data, the pluses and the minuses. If you did the advanced one, I gave you the data and you had to derive the pathway, with the caveat, as I said yesterday, that I wouldn't expect you to derive this entire pathway. So one thing that I do want to point out, especially if you did the mainstream one, it's pretty obvious if you're talking about like a sepia eye color, that it would be this enzyme 3 that's non-functional. And if you're talking about a scarlet eye color, it's this enzyme 5 that's non-functional. If you're talking about a white, though, it's actually this protein 6 that's non-functional. So my hunch is a lot of people said it would be enzyme 1 and enzyme 5, or enzyme 5 and enzymes 2, 3, and 4, which certainly is an answer, but generally we find with genetics, actually with most things, that the simplest answer is actually the correct one. It would be really unusual for there to be 2 non-functional enzymes in this pathway. It would be highly unlikely to find 4 non-functional enzymes. But if just this protein 6 is non-functional, then that means the proteins are actually getting made. They're just not being transported to the eye to show up as an eye pigment. And so that's the one for the white. So having both of these here, it's pretty easy to see what's going on. But I'm going to go over this data specifically with the sepia and scarlet again, and how you would derive these pathways. So we can see here, I don't know that I'm actually going to need to use a pen, but we'll see. So we can see here that molecule A turns into molecule B. Now, I wouldn't expect you to know that if you didn't have the pathway in front of you. So if you didn't have that, like I said, I wouldn't expect you to have that. I also wouldn't expect you on the advanced to know about protein 6. So that's also fine. But then we see that molecule B, when all enzymes are functional, you end up with equal parts yellow, orange, and blue. Okay? However, we can see in this data here that when we took out the orange, right, so we took out the orange, meaning we're crossing that out, and then we actually get more blue and yellow. Well, that makes sense now, because now you've got 50% going to yellow and 50% going to blue, whereas before you had 33% of molecule B. But the brown isn't actually affected. That's the same amount as before. So that gives you your first clue that that brown is on a separate pathway. So then your second clue comes from the scarlet. In the scarlet, we see that we've taken out the brown here. So if we are to knock that out, then we have our scarlet eye color with no brown. And we see, though, that the blue, yellow, and orange have not been affected. So again, that's still got to be the same pathway, 33% for each one, and that's not affected by the brown being knocked out because that's a separate pathway. And so that's what's giving us our clue that there are two pathways. The fact that in one pathway, knocking that out doesn't affect the others. And then we can see that those three, though, are on the same pathway, right? It's not 4 separate pathways or 3 separate pathways. It's got to be 2. And these 3 have to be connected because knocking out one affected the other 2. And so what I would expect you, if you did the advance, to have in your pathway, I would have expected you to be able to understand that there's this pathway here and that there's this pathway there. If you got that, then you really analyzed the data well and were able to come up with and derive what that pathway looked like. Hopefully then you can also see, if you did the mainstream, how you would have combined the data that you got in the first part. So the first part asked you to say something like, show which enzymes would be knocked out for each of the different colors. So hopefully you did something like this for the scarlet. I think I said to label it like SC, I'm not sure. So you did something like that for scarlet. And then you could see, okay, if I have that, then this has got to be a minus. There's got to be less brown pigment. Whereas these all remain the same. So those are the same. Plus means the same amount. It doesn't mean a little more. So if you look at the key, it says that plus means the same amount. Yeah. And then so the same thing, if you had something like this for sepia, and again, I don't remember exactly what I said to make it, maybe SE, that's what I'm going to say, right? If you did this on the first part, let me get rid of this. So if you had that SE, then you could see, whoops, wrong one, that, okay, there's going to be the same amount of brown that's not affected, so that's where you would have the plus here. There's going to be less orange, so that gives you the minus there, but now you should understand why there's more yellow and blue. So we do the plus, 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 plus. And then obviously white, there's less pigment than everything for wild type, so that's just all minus because there's no pigment in the eye at all. So hopefully this helps you better understand what we're looking at with more complex pathways, how you would have reasoned through to find the answers, and that this is actually something we can do. This is actually real. So obviously reach out with any questions you have if you want to talk about this more, but hopefully you find this informative. Have a good day.